Hello everyone. Today, I will present findings from Professor Liyan's group at Anhui University, published in iMetaOmics, titled Resistome Flow in Global Landfill Systems. In 2022, the WHO, FAO, UNEP, and WOAH highlighted antimicrobial resistance as a growing threat to public health. Landfills, key but understudied terrestrial systems, act as convergence points for ARGs. From landfills, these determinants may disseminate to surrounding environments via aerosol emission, groundwater impacts, occupational exposure, wildlife movement, and waste disturbance during excavation. Yet, compared with soils, wastewater systems, and freshwater environments, the global landfill resistome remains poorly characterized. This gap reflects both limited attention to landfills, often located at urban margins, and practical barriers to sampling deep, heterogeneous waste, which together have constrained progress in landfill resistome research. In this study, we found that multidrug-resistant genes dominate landfill resistomes, accounting for 33% of detected ARGs. We further assessed resistome mobility and observed that ARG density on plasmids was 3.5-fold higher than on chromosomal sequences, and that the number of plasmid-borne ARG-carrying contigs was 5-fold greater than in water systems. Finally, we delineated three major resistome transmission routes: inflow to landfills, internal transport within landfills, and outflow from landfill systems. We defined the landfill system as comprising 5 components throughout the municipal solid waste life cycle: solid waste, air, leachate, bioreactors for waste decomposition with leachate recirculation, and activated sludge and leachate treatment facilities. Globally, we annotated 40,442 ARGs, covering 22 types and 428 subtypes. ARG density refers to the number of ARGs per 100 kb of sequence. Although chromosomes carried a higher number and abundance of ARGs, plasmids exhibited a 6-fold higher ARG density than chromosomes. Furthermore, while chromosomes carried 2 to 3 times more ARGs and mobile genetic elements than plasmids, their length is to 100 times greater, indicating a higher potential for plasmids to carry ARGs per unit length. Landfill systems function as both major sources and sinks for environmental resistomes. Among surrounding habitats, adjacent soils enriched in ARGs and multidrug-resistant genes exhibited the strongest interaction with landfills and contributed 35% of detected multidrug-resistant genes. Notably, the high transport potential gas and liquid phases appears to facilitate ARG dispersal. Nearly 60% of multidrug-resistant genes detected in landfill air and leachate were shared between these two phases, indicating substantial cross-compartment exchange. We identified hosts of ARGs using metagenome-assembled genomes, recovering 1,537 non-redundant high-quality genomes. Pseudomonadota constituted 30% of the recovered genomes and carried 48% of all ARGs and 56% of multidrug-resistant genes. Additionally, we identified 6 pathogens, primarily distributed in solid waste and bioreactor samples. As Pseudomonadota are major ARG hosts, and our prior research identified Pseudomonas as a keystone taxon in landfills performing vital ecological functions, we selected Pseudomonas as a model strain. Analyzing 35 high-quality Pseudomonas genomes, we found that Pseudomonas aeruginosa harbored higher abundances of mobile genetic elements, virulencers, and multidrug-resistant genes compared to other Pseudomonas strains. We detected 428 ARG subtypes in landfill systems, approximately twice the number reported for acid mine drainage. The relative abundance of multidrug-resistant genes in landfills was 50% of that observed in freshwater and soil environments. In terms of genomic partitioning, the plasmid-to-chromosome ARG ratio was 1.1 in pig gut and 0.8 in cat, substantially higher than the 0.34 observed in landfills. Notably, despite this lower plasmid-to-chromosome ratio, the number of ARG-carrying plasmid contigs in landfills was 5-fold greater than that in global wastewater systems. Landfills are crucial for waste management and public health, but the movement of antibiotic resistance genes within and from them is a major concern, as they serve as significant reservoirs. Waste also attracts scavengers that can serve as vectors, potentially facilitating resistome dissemination. These risks may be amplified at open or temporary dumps, common in many low- and middle-income countries often located near population centers, where barriers to human and animal contact are limited and exposure pathways are expanded. A functional One Health response will require equitable investment, coordinated governance, and alignment of waste, water, and health policies to reduce antimicrobial resistance risks.